So now it's time to talk about gene expression, or how genes actually control the production of proteins. Nucleic acids. Before we dive in, we need to talk briefly about general nucleic acids, like what is the difference between DNA and RNA, because we've been talking a lot about DNA and we explained how it's double-stranded, two sides anti-parallel, blah, 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 but we have not really talked about RNA. So RNA, like DNA, is a molecule that consists of strings of nucleotides. But there are three basic differences between RNA and DNA. First off, the gray-blue backbone here of this RNA molecule, that's colored that way by the artist because it's using ribose as its sugar and not deoxyribose, which is what's in DNA. This is double-stranded DNA, and this dark backbone ribbon going both sides is basically based on sugars and phosphates, where the sugars are deoxyribose. Doesn't sound like a big difference, but ribose has one more hydrogen bonding site that deoxyribose does not have, and that's actually kind of a world of difference when it comes to chemistry and enzymes. The second difference between DNA and RNA is that RNA is single-stranded. It still twists like a half of a DNA molecule, and so it's not really straight the way we draw it often in class, but DNA is double-stranded like this. The other subtle difference is while the basic nitrogen bases are the same, the pyrimidine cytosine is the same, the purine guanine is the same, the purine adenine is the same. The pyrimidine for thymine is not the same in RNA. It's not thymine, it's uracil. Now, if you'll notice, the uracil is here and the thymine is here. And the only difference between the two is this little methyl group right there. A methyl group is a carbon and several hydrogens attached together, and this uracil is missing the methyl group right there. Methyl groups can't hydrogen bond, so it's not a big hydrogen bonding deal, but it does shape the molecule a little differently, and so as a result, enzymes can distinguish uracil from thymine. What's more easily distinguished, though, is ribose from deoxyribose. So, as it turns out, RNA cytosine can bind to DNA guanine. RNA guanine can bind to RNA guanine can bind to DNA cytosine, and RNA adenine can bind to DNA thymine. RNA uracil can bind to DNA adenine. RNA comes in three flavors. Let's go ahead and scroll down here a little bit and bring this up. All RNA is actually made of the same single-stranded, uracil-containing, ribose backboned nucleotides, but it turns out there are three or four, or actually more than that, but three basic types of functional RNA. So RNA differs basically a little bit in shape and majorly in function. Messenger RNA is the most common you hear people talk about. The messenger RNA's job is to carry genetic messages from chromosomes out to ribosomes so ribosomes can make proteins. Messenger RNA, though, is only part of the picture of protein production. Transfer RNAs, unlike messenger RNA, which are basically, they're not actually linear, but they are ribbony linear, like you saw in the last graphic. Transfer RNAs are not. Transfer RNAs are sort of shaped like a clover leaf, although in reality not exactly like a clover leaf, but they have four regions that actually are significant to their structure, and so that's why you see this is the anticodon region. These arms here are important for the ribosomes to grab onto, and the top is the amino acid attachment site. So transfer RNAs are shaped like clover leaves or T's or crosses. And then ribosomal RNA, this purple stuff here, is ribosomal RNA, and that's actually important to the structure of the ribosome and part of the function of the ribosome. And then those are the three big RNAs: messenger RNA, transfer RNA, ribosomal RNA. But there is one more, interference RNA, or RNA interference, RNAi, is actually manufactured during transcription, and it's actually used to control DNA activity. But we'll talk more about that at a later time. So that is the three kinds of RNA that are important to the functional aspect of production of proteins. The general flow of information in biology is that DNA information flows to RNA first, and the RNA then information flows into an amino acid sequence in a protein. So this is DNA. DNA information is coded into RNA lettered information, A, G, C's, and U's. And the A's, G's, C's, and U's determine the valine, leucine, isoleucine, tryptophan, phenylalanine, arginine, all those amino acid sequences in proteins. Except for weird things like retroviruses, this is the basic flow of information in biology, which brings us to actual gene expression. So as it turns out, all of your DNA in your cells doesn't actually transcribe and translate protein all the time. If it did, you wouldn't be able to have different cells in your body, and that would be a disaster. So really, this is quite complicated, but the actual mechanism of protein production is pretty similar from gene to gene, but I don't want to give you the idea that all of your DNA is always transcribing protein. Only some of the genes in any cell in the human body are transcribing proteins, and liver cells tend to transcribe and translate different proteins than kidney cells. And by the way, I shouldn't say transcribe proteins, it's transcribe and translate proteins. It's not just transcribe. So transcription is the beginning of gene expression. It is the production of RNA. And basically, transcription begins when RNA polymerase, which is RNAP here, binds to a sequence of nucleotides on the DNA called a promoter. So this is called like a promoter region here. And there are binders. These are proteins here that help to bind RNA polymerase to the DNA. These are really important. If there are no binders, RNA polymerase attachment to promoter is kind of slow, and gene expression activity is limited. But the enhancing proteins that are attached next to the promoters can really facilitate RNA polymerase attachment to DNA and speed things up. Basically, RNA polymerase opens up the two strands of the DNA all by itself, and then it actually copies one side of the DNA, moving from the 3' to 5' direction, going one nucleotide at a time. It matches A RNA nucleotides to T DNA nucleotides. It matches U RNA nucleotides to A DNA nucleotides, and G RNA nucleotides to C DNA nucleotides, and C RNA nucleotides to G DNA nucleotides. So it moves along following the rules for purine with pyrimidine, et cetera, et cetera. RNA polymerase, again, only works in the 3' to 5' direction, but remember, nucleotides don't fit together parallel to each other. They're anti-parallel. So when the RNA is made, it's actually made in the 5'3" direction. You'll see that in the animations coming up. Ultimately, when the RNA polymerase reaches a termination signal on the DNA, it will release from the DNA. And at that point, what you end up with is a piece of RNA, which is this blue string here. And you'll notice this is the beginning of the RNA where the 5' end is, because it's opposite to the DNA. And this is the 3' end. So the RNA polymerase started here. It went down the line. It put little RNA nucleotides together opposite to the DNA, and then hit a termination signal. Before this RNA can get out of the nucleus, three basic things can happen to it, or should happen to it. Let's go ahead and erase this stuff, and let's scroll down a little bit. The three basic things are, whoops, got to scroll back. Let's see, let's go back down. All right, I guess we had a refresh when I was scrolling, so here we are. RNA processing occurs. And so what RNA processing is, RNA processing occurs after this blue strand is manufactured. A tail is put on. It's called the poly A tail. That's put on the 3' end. That'll be here. This will be a poly A tail sticking out. It's at least 200 nucleotides long. It's to protect the RNA once it gets out in the cytoplasm from the cell's own RNA destructive enzymes. All cells manufacture RNA destroying enzymes to stop viruses from, like retroviruses, from hurting them. In addition, this side gets a special cap on it called a 7-methylguanosine cap. It's a 7-mg cap added to this end. And then lastly, the RNA is actually looped around like this. So you'll have a 7-mg cap on one side and the poly A tail sticking out. And a special spliceosome enzyme system will cut out like this section completely and shorten the RNA to a much shorter piece. This actually can become then RNAi, or other RNA molecules that are used to control the activity of the DNA that's in the nucleus. So it turns out the intron is this section of the DNA. This is called an intron. This section of the RNA. This is called an intron, the part that's cut out, and that is used to control DNA. Introns generally are non-coding regions of RNA that are removed before the RNA leaves the nucleus. They are important to DNA control systems, probably RNAi. So at any rate, 3 basic things happen to the RNA before it can get out. 1, the poly A tail is put on, that's here. 2, the 7-mg cap is added. And 3, the RNA is actually looped around and sections are cut out and the RNA is shortened. Now that brings us to the actual process of translation. When the RNA actually gets out to the cytoplasm, it actually goes or it bounces around, and eventually a ribosome will attach to the 7-methylguanosine cap end. And at that point, then translation will start. The basic sequence of steps is the ribosome is actually made of 2 pieces. And the small piece will attach first, and then followed by some transfer RNAs, and then the large piece. Before I can go into that, though, I've got to explain how the code is actually spelled out in the RNA. While DNA really doesn't have codons, RNA does. Every 3 nucleotides after the 7-mg cap is called a codon. Okay? Those are 3 nucleotide words that stand for amino acids. It turns out that these RNA codons each specify amino acids. Now, it turns out that there are a lot of redundant codons in the genetic code. So this codon wheel is basically a chart that indicates what codons are responsible for representing and dictating which amino acids go in proteins. So you'll notice this chart's read from the inside out. So an RNA codon reading GGG stands for glycine. But also GGA stands for glycine, GGC stands for glycine, and GGU stands for glycine. That's called genetic redundancy. So it turns out there's only 20 amino acids, but there's 64 different potential codons. In reality, there's only 61 codons. 3 of the codons they're here. 1, 2, 3. The codons UGA, UAG, UAA do not stand for amino acids at all. They are the stop signals of translation. The rest of the letters of the possible letters of G's, C's, A's, and U's all represent amino acids. And you'll notice that in rare cases like AUG, AUG only represents methionine. But on the other hand, ACU, ACC, ACA, and ACG represents threonine. Some amino acids are represented by up to 6 codons. Other amino acids, like tryptophan, are only represented with 1. It varies. And so there is some redundancy for some amino acids. For other amino acids, there is not. So Marshall Nirenberg got the Nobel Prize for figuring this out. He published in 1965. Marshall Nirenberg and Heinrich Mattei for their work on figuring out this codon wheel. The codon wheel is kind of a triumph of science. So that brings us now to the actual, translation process. And you scroll back up here. We got to talk a little bit about tRNAs and stuff. It turns out that tRNA molecules all have the same general shape. One side holds amino acids, and the other side is responsible for plugging onto the mRNA. The plugging side is called the anticodon. Each tRNA basically differs from other tRNAs by the anticodon, and there are enzymes running around in the cytoplasm putting amino acids, the correct amino acids, onto the correct tRNAs they belong on. That's called acylation. So there are acylation enzymes in the cytoplasm. There are 61 different acylation enzymes for putting on amino acids to the correct transfer RNA molecules. Now, translation starts when the mRNA molecule binds to the small subunit, and then a tRNA that's carrying methionine will bond next. The ribosome subunit then moves downstream a couple of codons to get in the correct reading frame, and then the large subunit binds. There are actually 3 basic sites where tRNAs can be found in the ribosome complex, but in most books they only mention the P site and the A site. There's actually an E site. I do not have in the animation the E site, but I'll show you where it is. The P site of a ribosome is where the very first AUG is bound, and then that is where the polypeptide is being held by the ribosome complex as translation takes place. The A site is the leading site of the ribosome where the new amino acids are being brought in by the new tRNAs that are coming down to bring in amino acids to the ribosome. And the E site is called the exit site. That's where tRNAs actually leave after they're used. So the polypeptide basically gets longer by a new tRNA binding to a new the next mRNA codon. The new amino acid then is brought in by that tRNA, and then a special enzyme called peptidyltransferase bonds the amino acid of the new tRNA to the amino acid that's already at the ribosome complex. And then translocation occurs, which means that the entire ribosome complex shifts down one codon to allow the next tRNA to bring the next amino acid. Eventually, protein synthesis will stop when a stop codon is reached on the mRNA. And that brings us then to the animations. And so what I need to do now is scroll down here and at least go to this animation. So let's go to the gene expression animation that we can actually control. So let's go to this one. And we need to go back there. We went wrong. Okay, here we go. So in fact, this went a little too far. And so let's get back here. Good. All right, so let's go back to gene expression. All right, make this a little bit bigger. All right, that's big enough. Scroll it up a little bit before we get started. All right, here we go. To animate this, I'm going to use the keyboard and let you watch how this works. This is a piece of DNA that's going to transcribe and translate some RNA. Now, the hand that you see on the left-hand side, this right here is RNA polymerase. Okay, it's the RNAP. And it's binding to the TAC here because that actually is right where the promoter would be right here. This is called upstream and this is called downstream. And the TAC is the start codon. So the hand is going to actually bond here at the TAC, and it's going to stick an A here, a U here, a G here, and it's going to keep going down the line, putting codons in position. This is the actual antisense strand. You'll notice it's actually making the other side, the AUG. See, the other side of the DNA is ATG. AUG is the same basically as ATG. So watch how the RNA polymerase looks. See, it's moving down. It's simply putting, now these are ribose sugars, and that's how the RNA is differing on the backbone from the DNA. So there goes the hand, it's moving down. It's bonding A to T, U with A, G with C, C with G, G with C, A with T. Do notice that AUG, CGA is the same as ATG, CGA. This is actually the coding side of the DNA, but it's not used by the RNA polymerase. The RNA polymerase is actually using this side, the antisense side, to make what is called the coding side, okay? So it goes down the line, zippity-doo-dah, and it puts all these RNA nucleotides together, and then it hits the stop signal at the end. At that point, it leaves. Right here, the 7-methylguanosine cap is put on the 5' end. This is anti-parallel to the DNA from which it was made. See, DNA is going 3 here to 5 over here. The messenger RNA is 3 here to 5 over here. So the messenger RNA always starts at the 5' side. So a 7mg cap is added here, poly A tails put here, and introns would be cut out of the inside of the middle of the RNA molecule. Then this goes out to the cytoplasm, and at this point, this is the small subunit attaching to the RNA at the first 2. It actually first attaches here, and then it shifts down 1 codon. Not 2, actually 1. And then the first tRNA comes in bearing methionine. And its anticodon UAC has to match AUG. Then the large subunit attaches. This is the P site right here. Here comes the next tRNA. This is the A site, okay? This anticodon plugs onto this codon, GCU is complementary to CGA, and arginine is carried by this tRNA. The peptidyl transferase reaction occurs right there, and then translocation occurs. The ribosome moves down. There's actually another site. There is as this moves down, there's an E site here, and I'll point out more importantly what it's going to do in just a second. But the A site is now open for the next tRNA to come in. It comes in, it's bearing histidine because its GUA anticodon matches CAU, and this anticodon tRNA carries histidine. Peptidyl transferase reaction occurs, translocation occurs. Now, this is actually the E site. It turns out that here is where the tRNAs are ejected after they're used, and it's here at the A site where the new ones keep coming in. So here comes one at the A site, peptidyl transferase, translocation. A new tRNA at the A site, cystine's in here, peptidyl transferase reaction, translocation shift. Okay, new tRNA, asparagine, new amino acid, peptidyl transferase, and then translocation. And this keeps happening until a stop codon is reached. This stop codon is recognized by, we think, the large subunit. At that point, everything's cut free. So this amino acid sequence is cut free. The mRNA goes to another ribosome to start another translation process. This sequence of methionine, arginine, histidine, threonine, cysteine, asparagine, tryptophan will now roll up into some kind of a ball, or it'll coil up or ziggy-zag or something, and it will form a structure that ends up being insulin, growth hormone, keratin, or collagen, or some other protein in the human body, and that's how proteins get made. So let's go back now to the notes and wrap this up. That's actual protein synthesis. So let's go back to the outline. That's what this was animating here. Kind of important, let's refresh the page for a second. We want to talk about decoding a gene. When you're decoding a gene, which you'll have to do on the AP test, let's get the pen selected here. When you're going to decode a gene, they're going to give you a piece of DNA. You have to note that as a student, you're going to read from the 3' side. This is 5' down here. This is 3'. You have to find the TAC. There's TAC. It's a start codon. So the RNA polymerase would start with the TAC, and it would transcribe AUG, CCC, GGG. Notice it's making the other side. This is the coding side of the DNA. That's what it's actually building. Let's see, it goes then I'm sorry, AAA, and then UUU, and then eventually it hits the stop codon here. ATT actually is a stop signal. It would manufacture UAA, and then it would be done. So it actually is making this piece of DNA right there, except all of the T's are replaced with U's. It actually, the RNA would read AUG, CCC, GGG, AAA, UUU, UAA, but the RNA polymerase actually, like a blind person reading Braille, it feels these nucleotides and puts those together. I mean, that's how it works. Then you have to translate. In order to translate, you got to look up the codons. AUG, CCC, GGG, and so on has to be translated at the codon wheel. So let's erase the drawings I've done. Let's go back to a cursor. Let's see, yep, there it is. And let's scroll up. So AUG, reading AUG stands for methionine, okay? One of the codons was GGG, G stands for glycine, C stands for proline, A stands for lysine, U stands for phenylalanine, UAA stands for stop. So the actual polypeptide that's made from this is methionine, proline, glycine, lysine, phenylalanine, and that's it. But do not write stop here. There is no stop. There's always one less amino acid than codons. So let me specify here. There would be 1 codon, 2 codons, 3 codons, 4 codons, 5 codons, 6 total codons, but there's only 5 amino acids, 1, 2, 3, 4, 5. Furthermore, all of the amino acids are bonded regimentally carboxyl to amino. So the carboxyl side of methionine is bonded to the amino group of proline. The carboxyl group of proline is bonded to the amino group of glycine. That's the peptidyl transferase reaction. A carboxyl group is C double bond O, bond OH, and an amino group is a nitrogen bonded to hydrogens. The way this works is an enzyme takes the HHO off there and makes a water, and then a covalent bond is made across from the nitrogen to the carbon, and you end up with a C double bond O, single bond N, single bond H. This is the peptide bond between a carboxyl group and an amino group. Every single one of the amino acids is bonded exactly the same way, and if you look through the sequence here, there's a backbone reading NCC, 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 NCC through the amino acids from the start to the finish. And methionine has its amino group sticking out on the left here, and phenylalanine has its carboxyl group sticking out on the right. The last thing to mention here is that a lot of times the Golgi apparatus will cut off some of these first amino acids to get rid of the methionine because the cell doesn't actually want those amino acids at the beginning there. They're a problem, so they're taken off. But they have to be there to make the protein because they're the start signal. But they're just cut off later to get the protein into functional shape. So what we're trying to say here is that this process of translation is responsible for all of your protein production and all your physical traits. Phenotypes, which is what our physical traits are determined by proteins. The skin color on these three men are all caused by the manufacture of different amounts of proteins, which causes different amounts of skin pigment, melanin, to be produced. And as a result, their skins have different color tones. Dark-skinned people simply have more enzymes for melanin production than light-skinned people, and melanin is for protecting humans from UV damage. So this is one of the thousands of, actually over 25,000 different proteins that the human body makes. Skin color is really easy to talk about because it's such a superficial trait. It has like nothing to do with your internal characteristics, or very little. It is not related to human intelligence. It is not related to stature or anything else. But it is related to protecting us from the sun. That's what melanin's for. And it turns out the lighter your skin is, the less protected you are from the sun.